Okay, good evening, everyone, and welcome. For those people who don't know me, my name is Professor Andy Nesbitt. I'm the Interim Dean for the Faculty of Engineering Sciences, and it's a great honour and privilege to be able to, well, one, welcome you all here today, both in person and online, and to say a few words of introduction to Professor Kasim Rafiq's inaugural lecture. We've got so many brilliant academics in the faculty. We thought it was really important we celebrate their successes. And because of that, we thought as a faculty, we would set up a more coordinated programme of inaugural lectures to celebrate when our academics achieve the promotion to professor. And we're delighted to have our very first one as Professor Rafiq, who's going to set a very high bar. So I'll just say a few words of introduction and a few thank yous, and then I'll pass over to Professor Rafiq. So Professor Rafiq is Professor of Bioprocess Engineering. He's also the Vice Dean for Health in the faculty and Director of the Institute of Healthcare Engineering. Now, Kasim has worked out that about 45% of our research is health-related, which shows the sheer scale of the role of that Vice Dean role. So it's an essential role for the faculty. Professor Rafiq is a chartered scientist, a chartered engineer. He's a fellow of the Higher Education Institute, and one of the recent accolades that Professor Rafiq has obtained was in 2020, and I'm going to read this out so I don't get this wrong. He was recognised as one of the top 20 influential and inspirational individuals in advanced medicine by the Medicine Maker. The Medicine Maker is a global publication that publishes success stories across the pharma industry internationally. So in that 20 individuals, there were Nobel Prize winners, Nobel laureates, chief executive offices of major multinational companies, and a number of distinguished scientists, of which Professor Rafiq quite rightly named amongst them. So a very prestigious accolade. Professor Rafiq's research is in cell and gene therapy bioprocessing, and again, we'll hear the details of that through the course of the inaugural lecture. We'll also hear some of the Professor Rafiq's career history. I'll just outline it really quickly. Undergraduate at UCL. We have all the best undergraduates as well. Then went to do a PhD at Loughborough University. Was then awarded a prestigious independent EPSCIRC Eton Landscape Fellowship to lead his own research activity. Was then appointed as a lecturer at Aston University. And then we were fortunate to recruit Kasim in 2017 as a lecturer. And so a very rapid and well-deserved promotion pathway to professor. We'll obviously hear about Professor Rafiq's research outputs, but I think it's worthwhile as well mentioning the full academic profile. So Professor Rafiq founded an MSc in manufacture and commercialization of stem cell and gene therapies. Again, apologies for reading that out so I didn't get it wrong, but that's the biochemical engineering's main master's program at our flagship UCL East campus. And Professor Rafiq has also been a major contributor to a number of institutional initiatives. So he led, and this is hot off the press, just announced last week, the Athena Swan Gold Award for the Department of Biochemical Engineering. So this is the third department in the faculty that's been awarded the Gold Award. There's only a handful of gold awards across the country. This is to promote women in STEM subjects, science and engineering. The fact, as a faculty, we've got 3 departments with gold is unprecedented across the country. But I mean, a real achievement as well. And again, I think it shows that as well as the research and education, the role that Professor Rafiq is having across the institute. I'm just going to say welcome to a few individuals in the audience. I think it's worthwhile just highlight a few people who've had key roles in Professor Rafiq's career. Arif Bobat, who's a secondary school friend, introduced Kasim to The Hobbit, and I think the reference there will become clear, hopefully, in the next 45 minutes. Dr. Eleanor Costa-Riol, who was Professor Rafiq's first PhD student, now graduated and working with Orchard Therapeutics. Dr. Sven Kille, who's the chair of the MSc Industrial Advisory Board, and who's travelled from Shrewsbury to attend this evening. Dr. Karen Koopman, who was one of Professor Rafiq's PhD supervisors. And then finally, although I am sure that all of Professor Rafiq's family have been integral in his career development and providing that support that is essential, I think I'd specifically like to mention the invaluable contribution to Professor Rafiq's career of his youngest daughter, Aisha Rafiq. So with that, I will hand over to Professor Rafiq and look forward to this inaugural lecture.
Fantastic. Thank you very much, Andy. Thank you very much, Andy, for those kind words, and thank you to all of you for being here today. For me, it's a huge privilege and an honor to be able to share my journey with you, but also share some of the research that we're doing at UCL and the research that I'm leading. For long, I've spoken to my kids, my children, my family, my wife about the work that I do, but they've never really had a chance to see it. So for me, this is a particular honor to share it with them, but also with all of you, because the one thing that I've always found with the research is that it's what drives the work that I do. And so hopefully by the end of this inaugural lecture, you'll be able to go on that journey with me and really get a chance to see why we do what we do and why it's so important. So that's really the focus of my goal today. And I'd like to thank Andy for that wonderful introduction and to all of those that are able to join as well. So just to kick off, really for me, the journey has been threefold. Looking at how we move from blood to bioreactor, but also a personal journey, how I've gone from being a UCL student to now a UCL professor, but also how we've moved away from looking at just addressing the scientific and clinical challenges to now thinking about the manufacturing and engineering challenges to be able to develop the therapies of tomorrow. And that's really the focus of my talk here today. So really there's 3 key areas I want to touch on. An overview of manufacturing new therapies, kind of a research overview. But as Andy alluded to, it's not just research that we do here at UCL as academics. There's a significant focus on training the next generation, but also a focus on citizenship and trying to look at things like gender equality and support and representation for minority ethnic academics, staff, colleagues, and students. And then finally, finishing on 3 final things, as you'll see at the end: choices, challenges, and collaborations. So what is it I do and why do I do it? We are now facing an unprecedented healthcare challenge globally. We have a number of diseases, diseases that we've never seen before that are now affecting as we go into our elderly ages. Things such as diabetes, Alzheimer, cancer. And the reality is we need to find a way to develop new therapies. The current existing treatments don't address the underlying issues. They don't address the problem. They simply address the symptoms of the disease rather than trying to cure the fundamental condition. And so this is really the goal. But what types of therapies can we develop? Well, the opportunity is there to now start thinking about cell and gene therapies, how we can take whole cells or start to edit and gene edit the cells so that we can develop new ways of targeting diseases and finding cures. And that's really the goal of my work. How do we try and use living human cells as therapies, as medicines that you can receive as a patient should you need it? Because the reality is, for some of the diseases that we're looking at, things like cancer, things like diabetes, things like Alzheimer's, we don't have existing treatments that work effectively, so we need to think of new ways. And this is where the opportunity arises for human cells as therapies. But what are the role of these particular therapies? Well, the therapeutic aim, what are we trying to achieve? We're trying to achieve a position where we can introduce either new genetic material to a cell so that it can correct genetic abnormalities, or to make a beneficial protein which can have a therapeutic effect. That's kind of the general goal. And ultimately, the way we intend to do this is to introduce cells or tissues into the body to address or generate a regenerative effect. And really, in many ways, we're starting to think now about the transformation of healthcare. How do we move away from a one-size-fits-all medicine where every single person receives the same pill, the same tablet, the same therapy, towards a more effective, safer, and more efficacious personalized medicine approach? That's really the goal that we're starting to think about. And the reality is we're heading in that direction, and we're very much there in certain ways with certain therapies. An excellent example is the idea of cell and gene therapies such as CAR-T, where we can take material from the patient, the individual who is sick, engineer that material in the lab, and then re-deliver that material back to the patient to treat a disease or an underlying condition. Now that sounds futuristic. It sounds like science fiction. How exactly can we do that, and what exactly can we achieve? But the reality is we are very much there. So I'd like to share with you, in this particular instance, the story of Emily Whitehead. So Emily, who you can see here, was suffering from acute lymphoblastic leukemia, basically an aggressive form of blood cancer. The normal treatment option for ALL, or acute lymphoblastic leukemia, is to have a bone marrow transplant. Emily received that and it failed. The second option is to receive multiple rounds of chemotherapy. And as you can see, Emily, with no hair due to the chemotherapy agent, she had received multiple rounds of chemotherapy, but the disease returned. And the third option was, well, there is no third option. Emily was destined to die of her disease because of the fact that there are no other options that we could think of. But at the time, and this is back in 2010, there was an experimental clinical trial taking place at the University of Pennsylvania, where they were looking at gene or cell therapies, the idea that we could take Emily's T cells and engineer them so that it could deliver a therapeutic effect, and that potentially her cancer could be cured. Now, this at the time was an experimental study, but you'll see the outcomes of that study with this particular video.
Patients that we're treating on this clinical trial have absolutely no other options left for them. These are patients who are unfortunately destined to die of their disease, and in a fairly short amount of time.
Emily Whitehead, 554.
So Emma is incredibly matter-of-fact about all of this stuff. This was a child who had had her leukemia come back twice. The parents were looking for a miracle.
What we've learned how to do is train the immune system to recognize and then kill tumor cells.
It's a procedure where we collect their T cells, and they're infected with a virus that will genetically change them so that they will now see and react against their leukemia cells.
And we actually use the HIV virus to do that.
So you're taking the HIV virus and infecting healthy cells with it to help kill cancer?
Yes. The virus has been engineered so that it can't cause disease anymore. But it still retains the ability to reprogram the immune system so that it will now attack cancer cells. We call those modified immune cells serial killer cells. The cell can kill more than 1,000 different tumor cells.
But the reality is the dramatic responses of cancer to new treatments are very unusual. We need to make it clear when we talk to a family that it may not work. Emma was given her T-cell treatment, and within a few days, she was very sick. She had breathing difficulties, she had blood pressure difficulties. We knew that she could not have gotten any sicker without actually dying.
But then a remarkable thing happened. The T cells were growing, they were starting to fight the cancer.
Within hours, Emma's fever disappeared.
It was like the calm after the storm. The clouds went away, and she woke up, and there was no leukemia. When that child survived, it was, of course, an amazing event.
So I love to show that particular video, and I show it to all of my students, whether they're undergrads, postgrads, researchers, colleagues, because it really highlights why we do what we do. We do it for this. We do it so that actually children who are destined to die of their disease don't have to face that fate or that outcome, not least at that part of their lives. So how can we start to develop these therapies moving forward? This is an image of Emily a few years later when she had the chance to meet the then president of the US, Barack Obama. And I've had the pleasure of meeting Emily and her family on a couple of occasions now, which is an absolute honor. And just to give you an idea of her character and her mentality, obviously meeting the president is a big deal, but you can't tell anyone about it. You know, there's a lot of security clearances and arrangements, so she couldn't tell her school where she was going that day, and she was going to miss a day of school. And so when the president asked her, you know, Emily, is there anything I can do for you? She replied with, as you can imagine, I need a note from you, Mr. President. And this is the note. Please excuse Emily from school. She was with me, signed Barack Obama. But I think that gives you an idea of the mentality of some of these cancer survivors. But it also shows why the work we do is so important. So we can have individuals like this who can lead normal, healthy lives, contribute to society, but don't have to live with the idea of dying in the relatively short term. So that's why we do what we do. And so just to give a bit more detail about this particular work, it's called chimeric antigen receptor T cells. That's very complicated. Basically, all it means is that we're now taking Emily's T cells, which are the cells that are meant to track and kill cancer, but are not working, or they don't recognize the cancer, and we're effectively integrating or putting in a new gene so that those cells can now recognize the cancer and kill the cancer when it's reinserted back into Emily. So we take the T cells from the blood, we generate these CAR T cells, so we've got some genetic code that allows us to integrate into the cell using a virus or a non-viral approach, and that cell now can target and kill cancer. We then grow the cells in a large amount, and then we infuse that back into the patient, and that cell can now target and kill the cancer cell. And in many cases, it works wonderfully well. But what's the drawback? You know, we've seen some of these opportunities. Well, when we start to think about the industrial or commercial context, until 2017, the US FDA, the Food and Drug Administration, the main, I would say, global regulator for therapeutics, had never before approved a cell or gene therapy. And now in 2024, we have over 40 approved cell and gene therapies. So these are therapies that now patients are potentially able to benefit from and able to access. And we start to see that actually the cell and gene therapy has the potential to be a multi-billion-dollar industry. So this is obviously a significant societal and health benefit, but it's a significant economic and commercial opportunity as well. And over the years, we've seen a number of large deals. For example, one of the key companies in the CAR-T space, Gilead, bought Kite Pharma, which was developing the CAR-Ts for $11.9 billion. And we've seen some other major large acquisitions and deals within this space. So what is the drawback? Why aren't these being rolled out across the board? Why aren't we developing these? Well, the drawback is this. They are significantly expensive. Most of these actually probably wouldn't get you a deposit in London, but you can start to see the cost of these therapies ranging from $200,000 for a stem cell therapy. This is one of the CAR T products that we just discussed, over $475,000 for a single infusion or for a single treatment. Now, this is potentially a cure, so when we start to extrapolate that over the lifetime of a patient, it's actually relatively low cost, but nevertheless, that's an eye-watering amount to spend on a single therapy, especially when other treatment options or other therapeutics are far lower. They may not be as effective, but they're far lower. So the challenge is why are they so expensive? Well, a key part of it is that the R&D and manufacturing process can account in some cases for over 50% of the entire cost of the therapy. So this isn't a scientific challenge. This isn't a clinical challenge. We know these therapies work. We've seen patients who receive these therapies and have been cured. Emily, to this day, and she received this over 15 years ago, is still healthy and cancer-free. We know these therapies work. But the challenge is now a manufacturing one, in addition to addressing the future scientific and clinical opportunities as well. So what are the challenges involved in manufacturing cells as living therapies? Well, I tried to boil this down into 5 key areas. The first is living cells are unpredictable. We're not dealing with chemicals where we can define and synthesize these precisely. They are unpredictable. Just as we are all unpredictable as living beings, cells are equally unpredictable, and therefore that presents a major challenge. We may have to grow them under different conditions. They may adapt to their environment very quickly, as we'll see. And we also have to think about the fact that these cells sometimes, if they're not provided with the right nutrients, which can be very expensive, they won't grow as we would like them to grow. The second aspect is both a blessing and a curse. So clearly we want to move towards more personalized, more targeted therapies. That's a huge blessing. But from a manufacturing perspective, that is a major challenge. How do we develop a manufacturing or therapy that is individualized to each and every patient? Imagine an iPhone that was manufactured to each and every person's hand, the size of their ear, manufactured their custom requirements. Whilst you might get some modifications, to truly personalize is a major challenge, and it limits the manufacturing scope and capability. The other key thing is strict safety and quality control. It's one thing manufacturing an iPhone. I mean, we saw some of the issues with Samsung and combustion inadvertently, and obviously those are major safety issues. But on the whole, there's a limited amount of quality control compared to what we have to experience when we're developing therapies that will go inside patients. We don't know how they'll react. We need to ensure that the safety profile is appropriate, but also the cells do what they're meant to do. The other key thing is they're sensitive to the environment. As living beings or living cells, they will react. As soon as the temperature changes, as soon as the pH changes, as soon as as they start to grow with other cells, they'll start to adapt and change. And that presents a major challenge. So we have to provide highly controlled conditions such as temperature, oxygen, nutrients to make sure that we can grow these cells effectively, because even small changes can affect their growth. And then finally, and this is always for me the biggest challenge, I work with a lot of clinical scientists, I work with a lot of development scientists, and they have some phenomenal, fantastic ideas and potential products that could be developed. But the challenge often is they don't think about the manufacture and the scale up early on. They will often think about all the scientific challenges, great, but the reality is if we want to get these to patients, we have to think very early on about how we're going to manufacture these therapies and get them to a scale and to a cost that can be accepted by wider society and by, in the UK, by NICE or the NHS, and in the US and elsewhere by private healthcare insurers. So scaling up is a huge challenge and something we need to think about very early on. But this isn't just me saying it. What I'd like to now show over the next couple of slides is that this is a global challenge. So back in 2018, the then commissioner of the FDA, so leading the FDA, Commissioner Scott Gottlieb, outlined that actually the major challenge or the greater challenge with cell and gene therapy is the limitation in manufacturing capability. Not the science, not the clinical outcomes, but the manufacturing capability. And he went further to outline that actually we need to start thinking about scaling manufacturing processes that retain the inherent quality attributes of these products. This was further reinforced very recently in 2022 by the CBER director. So this is a division under the FDA that regulates these particular therapies. So Peter Marks, the CBER director, outlined that we should start thinking about, or we should be considering, automated manufacturing as another solution. So really, this then comes to the thrust of my research and why we do what we do and what are we doing. So what are we doing at UCL and in my team to improve cell and gene therapy manufacturing processes? Well, really, it comes down to 3 key areas that we've identified. The first is automation, so starting to think about how we can automate processes and what we mean by automation. I won't go into that whole philosophical discussion now, but I'll show some examples of how we're leading in terms of automating processes, of developing automated technologies. The other key area is bioprocess optimization. How can we go from these small-scale manual flasks through to actual bioreactors that will allow us to start thinking about scale? That is a challenge, but one that we're very keen to address and very well placed to address. And then finally, as with the rest of the world, we're looking at how we can start to integrate digital tools such as AI, machine learning, and process modeling into some of these manufacture of these therapies or the development of these therapies. And that's through extensive collaborations we have within the department and outside of the department as well. So I'll touch on all 3 of these areas throughout the course of my talk. So the first is automation strategies for cell and gene therapy. So during my PhD and my fellowship, when I was at Loughborough University, we had the opportunity to work very closely with a company called TAP Biosystems, which got acquired by Sartorius. And they had a microbioreactor platform. So this is a platform that allows you to grow cells in effectively a tic-tac box shaped device. And this is the actual vessel that you can grow the cells in. And so within this platform, you can potentially have 24 or 48 bioreactors in a single platform that is controlled by a robotic arm. Now, this was a platform developed by the team, but it had never been used for cells that are adherent. So some cells, like blood cells, can grow in your blood and without a surface in order to grow. So they can just roam around in your body, no issues whatsoever. Other cells, however, are adherent. So they need a surface in order to grow. They need to stick and attach in order to be able to grow. Otherwise, they'll die. So this platform had never been devised or developed for that particular application. And so we worked very closely with the team to be able to adapt the platform and develop processes that allow us to get good growth within these bioreactors. I'm not going to go through all the details of the science. I don't think that's particularly relevant here, but if you are interested, we published it in that paper at the bottom there. But following the success of that particular project, so that was a project we started in 2015. Following the success, I had coffee by chance with one of the colleagues from Sartorius who mentioned and knew about some of the work that we had done for developing or supporting the development of this for adherent cells. And they said, look, we've got a major challenge. We've got the larger scale platform, which is this one here. This one here. The size of a Coke can, about 250 milliliters in scale. We've got this vessel, but the problem is that, again, it's great for suspension cells, but it's not good for adherent cells, cells that need a surface in order to grow. Can you help us design a new vessel that can allow us to achieve that? And the answer was yes. So we were able to do lots of both experimental work as well as modeling work to be able to deliver this. And this was a huge success. I mean, I think we estimated, and we got funding for 2 years for the project. I think with all of these things, you're always optimistic at the beginning, and then you think, can we do this in time? We actually did this within 18 months. And in the final 6 months of the project, Sartorius used this to market the vessel, and it's now doing very successfully. I wish I'd taken rights on it, but it's now doing very well at Sartorius and was the feature of a ref impact case study, which is a university metric as part of the last research excellence framework. And this is the actual vessel that we designed. So we couldn't change the outside. The outside had to be the same because it had to slot in, but we were able to change the engineering parameters within the vessel to make it better for growth, changing the impeller, changing the number of impellers, removing baffles and a number of other aspects to improve the production and growth within this particular vessel. And they now market it on their website as a cell and gene therapy vessel. So those are 2 examples of kind of within an automated context for process development. But what about automation for manufacture? So when I was at Aston University, I was part of a European project working with a number of fantastic international and European collaborators as part of a project called AutoStem. So this was a €6 million project to look at how we could develop a completely automated platform where we could isolate stem cells from a donor. We could then grow that in a bioreactor in a completely automated platform or system and then deliver that back to the patient. So the idea being that actually from start to finish, it's a completely automated process. So this was the initial scheme or the CAD design. And again, this was a 3-year project. And I genuinely remember the first consortium meeting in Dublin thinking, This is very ambitious. I don't know if we'll ever get there. But let's give it our best shot. And our focus was very much on the bioreactor, so that was our main goal. But working very closely with our partners at IPT Fraunhofer in Germany, who are phenomenal. So this was the original CAD design. And the answer was we were able to get there within 3 years. And on a couple of slides, I'll show a couple of videos of it in operation. But what we could show is that this was growth of the cells. So the cells are attached to these beads. These large circles are beads or microcarriers, surfaces that the cells can attach to in order to grow. And you start to see, as the cells start to grow on these microcarriers, they form these kind of web-like structures. And then they start to grow and more and more and more on these microcarrier beads. And we can then extract the cells from the microcarriers once we've actually allowed them to grow. So this was some of the growth data. I won't go through all of this. Again, this is published. But the main thing was, compared to our manual and automated process, there was very little difference in terms of the overall outcome. So we could show that with our automated process, we could remove the human operator and have this in a completely closed automated platform. And here's the platform in operation itself. So this was developed by our colleagues over in Germany, and we were part of the design of this. So this is at the end of the process, where the robotic platform is now adding the cell media and the cell reagents to a vial, and then transferring that, and that will then go into cryopreservation and freezing. The next video is then of the bioreactor itself. So this is the work that we were leading on to optimize the production and growth. So whilst we have one donor or patient, a production batch, we are manufacturing a second in parallel. And there's been lots of conversations with the regulator how we can achieve that to avoid cross-contamination, but also enable effective manufacture. But for me, this is kind of automation 1.0. It's very much the automation of just removing the human operator from the process and just having a device or a robot be able to do what the human does. Now that's great, because as humans, on a Monday morning, if we're working in the lab, I've been in the lab myself on a Monday morning many years ago now, but I've been in the lab for a number of hours, and I know that on a Monday morning, it's very different to a Friday evening when you're kind of looking at your watch and wanting to go home, or in my case, with my wife waiting for me in the car when I was doing my PhD, and me having to then quickly rush because I told her 10 minutes, it's taken about an hour and a half, and yeah. So that's really the goal. That's one of the goals of automation, is to remove the human operator and that variability that is associated with humans. But we also want to start thinking about how we can automate decision-making and how we can automate process development, so that we can start to think about not just the physical activities are being automated, but also some of the process activities or decision-making and next steps are automated as well. So the second research focus is very much on bioprocess optimization and control for cell and gene therapies. Now, I apologize to many of my researchers, because there were literally hundreds of slides I could have included in this. And actually, a lot of today was deciding what do I include, what do I not include, what is the narrative, and so on. So I just thought I'd focus it on the CAR-T work, because there's so much we're doing in viral vector. So we do have a huge amount of work in the viral vector. So why are we manufacturing virus? The virus, in this instance, is our delivery vehicle. So it's what we're using to deliver that genetic material to the cell. It acts, that's what viruses effectively do. When we get sick with a viral infection, it's basically a virus that's entering our cell, changing our genetic material, inserting its own, and then that being replicated again and again and again. What we're doing is removing the harmful material and inserting it with the material that we want it to do. It's basically a more elaborate postman, effectively delivering a parcel. So that's very much the viral vector part. I won't focus on that now, but there are two main viral vectors that we use, lentivirus and adeno-associated virus. I won't touch on that work, so apologies to my researchers for not touching on that. But that is some phenomenal work that we're doing in that space. But what I really want to focus on is on the cell manufacturing aspects. Once we've edited the cells, how do we then manufacture those cells? And for me, it comes down to moving towards bioreactors. This is a very simplistic way of putting it, but in reality, it's not that challenging from a conceptual perspective. How do we move away from manual processes where humans are having to do this again and again, where they'll make mistakes, but more importantly, where there'll be a lot of variability, there's a lack of control, but also a lack of scalability, to a platform that is truly scalable, that has sensors and integrated process analytics to allow us to be able to automate that decision making. So one of the key platforms I've used throughout my career is a stirred tank bioreactor. So as Andy mentioned at the beginning, I was a UCL undergrad student in the Department of Biochemical Engineering. And all of our focus, as you would expect as a biochemical engineer, in terms of large-scale upstream or cell manufacture, is with stirred tanks. But you come to clinicians and scientists who are working with T cells, and they all think their cells are special. These cells can't be agitated, they can't be stirred, they are special. And then when you challenge them and say, well, actually, these cells grow very effectively in the blood, they often say, but imagine the shear forces, or when you're agitating at large speeds, that's going to damage the cells. These cells don't have a cell wall, they're going to get damaged. And then again, you go back to that point, they grow very effectively in the blood, where the shear stresses are far higher than anything they would experience in stirred tanks. But the other benefit of stirred tank bioreactors, and there's numerous ones, but the other big one is we can scale these bioreactors up to 20,000 litres. There are products, not cell and gene therapies, but other pharmaceutical or vaccine products, which are being generated at 20,000 litres and beyond. So these are platforms that are fantastically designed for scale, for production. The regulators know how to regulate such processes. They've been familiar for years with these types of platforms. So why don't we use them for T cells and CAR T production? So one of our first research questions when we started this work, and this was work led by Elena Costariol, my first graduated PhD student in this space, was how can we grow CAR T cells in a stirred tank bioreactor, and how do they compare to growth in T flasks? Well, the answer was actually we can grow them very, very well, and actually the best growth, so if you see here at the end, the red line, the green line, the purple line, and so on, are all bunched here at the end. We were getting over 4 million cells per milliliter. So per milliliter of solution, we had 4 million cells. And you can see actually the higher we took the agitation rate, the better the cells grew. When you compare that to the static T flask, which is what often many scientists would grow them in, actually it was the lowest level of growth that we were achieving. So we knew we could grow these cells in these stirred tank bioreactors. But that was at the small scale, kind of this sort of scale, the 250 milliliter scale. The question was, could we now do this at the liter scale? And the answer very much again was yes. So moving from this bioreactor, the 15 milliliter, through to the 250 milliliter, and up to a liter universal platform, we demonstrated what, for me, was the most important finding. Regardless of whichever platform you used, we got identical or very comparable growth. So we could do all of our development in this platform where it's very, I wouldn't say cheap, but it's much cheaper to use this platform because you're using less volume, less media, which is very expensive, less growth factors and so on, and be confident that the more you develop and optimize your process there, you're confident that actually at the larger scales, you'll get the same outcome. So you have a small scale model that gives you an idea of what things would look like at the larger scale. So that was great. Our next research question was, would perfusion allow us to achieve even higher concentrations of cells? So what is perfusion, before jumping into the detail? Perfusion is effectively this. So this is the same Amber 250 bioreactor, but we now have this contraption. What does this contraption actually do? Well, what it does is cells need to be replenished with media. It's like, like any living being, no one wants to live in their own excrement or waste or filth, but also they need to be replenished with new nutrients, new growth factors and so on in order to continue growing at their optimal. And what this perfusion device does, which is connected to this bioreactor, is that it extracts out the waste or whilst retaining the cells in a filter, and then it replenishes it with new media. So you're now basically, in a nutshell, feeding the cells with what they need and removing it from what they don't need. It's like my children when they were younger. I would often have to do, not that often, my wife did most of it, but nappy duty. So you want to make sure that you remove the waste, but also make sure that you feed the cells. That's effectively what we're doing with this device. So you can see here the flow paths. I won't go into all of that. But the challenge is that this, again, hadn't been used for CAR T production. And we actually showed, so in our previous process, which we call fed batch, which is when we're just adding media, we're not removing any of the waste, you can see our production was relatively low. But when we started to move to perfusion, we saw even higher levels of growth, better than anything we'd ever seen before. You might be thinking, why is there so much difference between these conditions? These were all perfusion conditions. Why was there so much difference? We're seeing these big variability. Well, actually, that was designed, because what we're looking at is, through what we call a technical term, design of experiments, we're actually changing multiple parameters in each of our experiments to identify what is the optimal sweet spot where we get the best growth and the best bang for our buck. And that's, in a nutshell, what we were trying to do. So this exercise was very important because it was showing us there were a number of conditions where it would be average, but then there was one particular sweet spot where we were getting significant levels of growth. So removing all the other lines, you can see from our best design of experiments perfusion run, now where we've got this device attached, we had 4.5 times more cells compared to the standard fed batch, where we didn't have any growth. We weren't renewing, replacing the media and removing the waste simultaneously. And so if you start to look at the numbers, our final T cell yield in 7 days with our fed batch on our perfusion was 4.5 times. So the number of doses we could therefore produce in that period was quadruple. But more importantly, when you're talking about patient-specific therapies, these patients don't often have long to live. This is the last treatment option that they're destined to receive, and so therefore they don't have particularly long to live. So the time to reach the first dose is significantly lower. And often a concern with perfusion is that you're wasting lots of media, which is very expensive. But actually we showed there wasn't a huge significant increase in the overall media consumption with these particular therapies. The other thing, and I put this slide here deliberately, I know most people won't understand it, but I wanted to show, because it's not just about the quantity of cells. One thing is the quantity. We want to generate lots of cells that ideally have the right characteristics. And this is what this graph is now showing, well, these graphs are now showing. Do the cells actually have that CAR transgene? So has that deep piece of RNA, DNA, or nucleic acid material, has it actually penetrated the cells? And are the cells now expressing that new marker that can target cancer? That's basically what these graphs are showing. So it's showing that upon harvest, we have our cells which are CD3 positive, that's a marker, but they're also CAR positive. So they're expressing, or they have on their surface, that particular receptor that can target the cancer. And we also looked at inoculation is when we put the cells into the bioreactor at the beginning of the process, and harvest is at the end of the process. And you can see at the beginning and at the end, there's very little difference in terms of the level of that gene expression. So they're still retaining that receptor at the end of the process. Again, I'm not going to bore you with all of this data here, but I've put this in because this is another important feature. So one thing is, do the cells actually have that CAR receptor? So can they be effective when they get in the body? The second thing is, and you'll find this quite funny, I did when I first learned about T cells and did a deep dive into immunology, T cells go through different states. So they will change the way that they react. And that's what happens naturally in our body. When there's a viral infection or when there's some sort of infection that the T cells need to respond to, in the very early stage, there are very few of them, and they're in what they call the naive state, kind of a very naive but highly effective state. And as they start to progress, start to grow in the body naturally when we respond to infections, and they start to attack the viral particles or the bacterial cells, they'll start to go through and change their state. So what we call their memory state. So they'll go from naive to central memory, effector memory, and effector. And the challenge is we don't want the cells to be in the effector memory or the effector stage. If they are, it means they're what we call terminally differentiated, and they're not going to be effective when they get in the body. And the great thing is that actually we saw the vast majority of our cells were either in their naive state, and this is kind of what we would expect, about 20%, and the vast majority were in the central memory state. So they're the cells that we actually want to deliver back to patients. And the other thing is something called exhaustion. So T cells get exhausted. They actually go through a process whereby they can no longer function. Once they grow to a certain level, they become exhausted and they don't want to grow anymore, they don't want to do their effect. And what we found was actually at the start of the process, at the inoculation, the T cells are expressing high levels of exhaustion. And that's because we're basically forcing them to grow. But as they start to grow and as they're producing this bioreactor process, because we're providing with the right nutrients and environment, actually the exhaustion when we remove them from the bioreactor is actually less than 5%, which is fantastic. So the T cells are not exhausted by the end of the process. So this is kind of some of the work that we're doing and we're continuing to do to start to look at how we can go from even higher numbers. So we just touched on there how we've gone from 4.5% or 4.5 times increase. We've actually now, in some of our most recent studies, even higher, where we've gone up to 9 times the increase. I didn't want to show all of that data. That other work has now been published, but I want to show some of this data where we're now actually 9 times even higher than where we are, and that continues to increase and improve. But this, for me, is really kind of the creme de la creme. It shows the T cells in action. So in this first video on the left here, you've got normal T cells. So these are not modified in any way. These are just normal T cells. They don't have the gene. And you have cancer cells in green. And what you'll start to see is that the T cells don't recognize the cancer cells. And the cancer cells continue to grow and grow and grow until effectively After about 48 hours, you get to a point where all you have in your culture is just cancer cells, because the cancer cells have continued to grow unrestricted and unchallenged. However, when we have our gene-modified T cells, so again, now in this case, our gene-modified T cells are the gray ones, and again, the green ones are the cancer cells. What you'll start to see is that actually the CAR T cells, which now have this gene, are able to recognize the cancer cells. They're able to surround them, hone in on them, and then basically neutralize and kill the cancer cells. So what you can see here in exactly the same time period, two very different stories. Unmodified T cells full of cancer, and then on this image here, or this video here, the CAR T cells which have surrounded the cancer cells and effectively neutralized them. So that shows the T cells in action, and these cells are basically in our labs just 100 yards from this lecture theatre where we're growing these and developing these processes. And then the final research focus, the process modeling, AI, and digital twins for cell and gene therapy manufacture. So this is driven by a question that we always ask ourselves in our group. What does the future of cell and gene therapy manufacture look like? And really for us, it comes down to this idea of AI, machine learning, process development. So we were very fortunate to be part of a successful consortium called AidPath, a European consortium, the Artificial Intelligence-Driven Decentralized Production for Advanced Therapies in the Hospital. Basically, all that means is using AI processes and manufacturing in the hospital next to the patient. And the idea was this was based on the success of our AutoSTEM project, so working again with our key partner, the Fraunhofer IPT, but also a number of other European and international partners, looking at how we could develop a completely optimized or decentralized process as part of the smart hospitals of the future. So as part of this platform, so we're developing this automated system. The project is in its final year, and the platform has been delivered to the hospital in Germany. I'm actually heading out there next month, as we want to do a promotional video and record the platform in action and demonstrate how we can generate not only automated production of T cells and CAR T cells, but now platforms and processes that are using AI process models and other AI integration in the hospital management system to generate these CAR T cells as well. So a huge shout out to my European collaborators, I think many of whom are watching online as well today. So within this particular process, or within this consortium, we've got two main goals. The work that I'm leading and that I'm responsible for is how we can start to think about developing a non-viral expansion process. All the work I showed before is how we can generate those CAR T cells, but we have to use a virus as the postman. The question here is actually, could we remove the virus and do it in a non-viral way and still get that material inside the cells? And so we can think about different ways of doing that, shocking the cells to create pores to allow the material to enter. And this is effectively what we're doing with an electroporation device. And this is working with a company called Lonza, who have a manufacturing bioreactor platform for single patient manufacture. And we've shown that we can both optimize the electroporation process so that we can create those holes in the cells using electricity and deliver the material, as well as then grow the cells subsequently as well. And then the final part that we're doing within this consortium is led by my colleague, Dr. Stephen Goldrick and Dr. Joe Egan, and we work very closely together. We provide a lot of the data for their models. Is starting to think now, can we use AI, machine learning, or advanced algorithms to start to predict the performance of cell growth very early on? In many instances, one of the challenges with personalized manufacture is that as you start to grow the cells, you often may end up in a scenario where the cells for that patient don't actually grow, or they don't generate enough cells. If you can learn that very early on or predict that, that could save a lot of time, a lot of effort, a lot of expense. And so here, it will be a video in just a second, but what you're seeing here is effectively this dotted blue line is the future at-line data. So this is what actually happened in real life, and the reason it's called the future at-line data is that we knew at the end of the process, we knew the values, so you can see what we're trying to get to. And this orange line this orange line here is what the model originally predicted. So you can see at day 0, the model was actually quite far off. But as we're collecting data in real time, so as the bioprocess is happening, you'll start to see that actually, as it starts to move, as we're collecting real-time data, the model is getting far more accurate. So within about 24 to 48 hours, we're able to effectively predict what will be the final cell number, how the cells will respond, and what we can generate. And that was based on 4 data sets. We're now in various projects where we have over 150 data sets where that will refine the model even further. So many thanks and credit goes to my colleague, Dr. Stephen Goldrick and Joe for leading on that work. So the final part of my presentation. So for me, manufacturing is only half the problem. We know there is a challenge to get these therapies to patients, but in order to do that, we have to start thinking about training. We have to start thinking about generating the next workforce. This was highlighted by the UK Cell and Gene Therapy in 2019. They highlighted that by 2024, which obviously we've now passed, the total number of jobs that would need to be filled was over 2,000 for bioprocessing and manufacture, and we're nowhere near that number. And that's presented a major challenge. And so the question we often think to ourselves is, what are we doing at UCL to address that challenge of skills development and generating the next leaders? And really it comes down to 2 or 3 key areas. The first is our new master's program. I say new, it's now 5 years old, but as of last year, it was started to be delivered from our new UCL East campus. So delighted to be able to lead on that. And now Dr. Rana Khalife is now the program director and taking it to new levels and to new heights. But also delighted that we have a number of industry and commercial partners who are supporting that development of future leaders and advising and mentoring some of our students. And I can see some of our alumni in the audience today, which is fantastic. But I want to make a particular shout out to our industry advisory board members in particular, because they are all busy, super busy. I mean, you ask anyone for their time, it's always a major constraint, as it is for all of us, I'm sure. And I remember approaching each of these individuals, and this was kind of our first choice list. You know, we wanted to kind of make sure that we could get there, and I was expecting genuinely most of them to say no, that we're too busy, we'd love to, but we don't have the time. Every single one of them said yes. And they realized, A, for 2 reasons. A, because they're super enthusiastic about training the next generation as well, but they also know the importance and the opportunity to recruit from this particular program. And that's something that there has now successfully occurred. So I'd like to shout out to all of them, in particular Sven, who chairs our industry advisory board and is here today. But also the other key area is training doctoral students. And we're delighted to work with Oxford Biomedica and University of Oxford in developing a program to train 24 doctoral students in the viral vaccine and the viral gene therapy space. And this is a successful program where we've now completed our cohort. We now have all 24 students, 8 in each year, who are on this program and hopefully enjoying it. Again, some of them in the audience today. So hopefully they're enjoying that program, and the idea is, again, from a doctoral standpoint, to be able to support the sector and the industry. But the other key thing is recognizing that we work within a higher education institution. And as Andy alluded to, I won't dwell on this, but very recently, I was fortunate enough to lead a fantastic team of colleagues as we put in our Athena Swan application. I would say this was probably more challenging to write than my own PhD thesis. It was about 180 pages long, but working with a number of wonderful colleagues, it was fantastic to get this over the line, and then delighted to be awarded the highest recognition of Athena Swan Gold. And I think that's really a testament to our entire department, to our staff, to our students, to our colleagues, who really all we did was capture their story and put that in the application and present that to the committee who were assessing, and that clearly came through. And as Andy alluded to, we're the only faculty across UCL to have 3 goals, the only faculty, as Andy alluded to, anywhere in the country, which is amazing. So many congratulations to chemical engineering, to computer science, and to our colleagues in biochemical engineering as well. And then finally, the last part of my presentation. This isn't the end, there's a couple more slides, but this for me is probably the most important, and it's the thank yous. It really is kind of my opportunity to thank all of my wonderful colleagues, mentors, friends who have been with me on this journey, as well as indeed my family as well. So really to start off with my UCL biochemical engineering academic colleagues, and from across the department. I can't name every single one, so I thought I'd put all the pictures on that we have of the slide, but to thank all of my colleagues, both the senior colleagues, the newer colleagues who've joined the department as well. It has been an absolute pleasure working with you, and we are truly like a family, and that's been a huge integral part of our success as a department. I'd also like to thank actually the professional services team, because often we as academics get all the limelight, get all the glory, and we deliver on the research or teaching, whatever it might be, but none of this would happen without our professional services colleagues. The individuals who take a lot of the brunt from academics, they They often have the challenge of having to work to tight deadlines because, you know, whether it's myself and Laura, apologies if I've sent you a last minute email to say, look, can we get this grant submitted or can we get the costings approved? But to be able to work with phenomenal colleagues who've been able to support all of us throughout has been a remarkable honour and I think a great opportunity and part of our success as a department. But I'd also like to thank a number of mentors. Some of us are in the room and some of us are unfortunately no longer with us. I'll start on the top left. That's my favourite picture of Professor Chris Hewitt and Professor Alvin Nino. So these were, in addition to Professor Karen Koopman, who I'll come on to in a second, but Professor Chris Hewitt and Alvin Nino were both part of my supervisory team. Chris was my primary supervisor and Alvin was my third supervisor and Karen was my second supervisor. But with Chris and Alvin, they were true giants of the field. Alvin has a huge history at UCL, left to go to Birmingham and then supported at Loughborough, and unfortunately both passed before their time. Chris, back in 2019, so he missed the pandemic, and I'm sure he would look down and comment on that and think, you know, I missed that. I think it was the hottest day of the year, as his wife Sandra commented on. So again, I think he would have been delighted to miss that, but also Alvin as well, true giants of the field, as those who know them, and had a huge impact on my career, but also on many others as well. I'd also like to particularly thank Karen, who's here in the audience today, who was also one of my supervisors of my PhD project at Loughborough University, as well as Professor David Williams, who, again, had an enormous impact and continues to have an enormous impact on the work that I do, in my mentality, the way I think about things, and I can't thank all of those colleagues enough for the work and the impact they've had on me. But I'd also like to thank a number of industry and academic colleagues in particular. So Sven, obviously I've touched on already, leads our industry advisory board, but does so much more than that and is a true leader of the field, as well as Dr. James Miskin. So Dr. James Miskin, unfortunately, couldn't be here today, but he led on a lot of the vaccine development and is also a co-director of that doctoral program we have with Oxford Biomedica. Two peers who are on the doctoral program with me, so Tom Heathman and Alex Linus. I still have regular calls. I think I caught up with Alex just last week. And Tom has been a huge inspiration for me, a role model, but also a dear friend and colleague. And then finally, Professor Chris Mason. Had it not been for Chris back in 2008 and his module, I probably would have ended up in something like investment banking, because that was kind of the trajectory I was heading on. I saw Lehman Brothers at the time needed biotechnology specialists, and it was not until I actually came across stem cells, regenerative medicine, that I realized the opportunity that we could play as engineers. So it was his inspiration and his passion that drove me to enter this field. I'd also like to play a huge shout out to the researchers and the funding agency. So my entire research group, most of them are listed here, some of them I couldn't get pictures of, but I'd like to thank all of them. None of this would happen without their input and support and their leadership. They make me look good. They do all the work, they do all the thinking, and I'm just here to let them shine. And that's really what I see as my goal, is to try and generate and nurture these phenomenal individuals, whether it be Pierre leading on the CAR-T work, or indeed Pedro, who's been with me for over 8 years. He's put up with me. He was my first PhD student at UCL and is now the lead researcher in my group, and a number of other colleagues who are in the room here today to celebrate this inaugural lecture, and the funding bodies and key industrial partners that have supported this work. The final thanks goes to my wonderful family. So they've been patient with me, they've supported me throughout my life. So this is a picture of my father and my mother and my kids as we were on holiday in Istanbul. So I'd like to thank them in particular for all of their wonderful love and support. My siblings, Farina Baji, Farhan Bhai and Farid Bhai, who have been I'm the youngest of 4. So they've again supported, sacrificed so that I could achieve what I could achieve. So I'd really like to thank them for all of their work. To my wonderful nephews and nieces, some of whom are in the audience today, and to all of my in-laws and relatives from all sides as well. But it wouldn't be remiss of me if I wasn't to include some videos of my children. This one particularly is a bit of a dagger in my heart, as you'll see why. So this is my daughter Miriam, who is now 17 years old and still scares me that I have a 17-year-old daughter. This was her when she was about that tall. This is a video of her.
Do you love Daddy?
Yeah, I love Daddy.
Do you love Mommy?
Yeah.
Do you love being here?
Yeah.
Who do you love the most?
I love Daddy. I love Daddy. I love Uncle Yusuf.
You can hear my disappointment as she says she loves her Uncle Yusuf right at the end. So that's always a dagger to my heart as she remembers that. Then there's my son Yahya, who...
So Daddy in the train.
talking about dropping me off at the train.
We really beat the little chain. No, we didn't. No, we didn't. Don't drop him out to uni.
And it's scary to think now he was that tall, 10 years ago. And I will finally acknowledge to him, we've been having this long debate, he's now actually taller than me. So I thought I'd save that for this moment. We've been having a long-running debate, I've been standing tip-toe, but he's now officially taller than me. And then finally, my youngest daughter, who was actually sick when we took this video. I was trying to get her young to try and compare.
Am I the best daddy in the world?
Yes.
I love you, Mike.
Positive encouragement, I want to make sure she's good. And that's you can tell she's the most theatrical out of all three of the kids as well. And then finally to my incredible wife, who's put up with me throughout all of these years, who waited hours to pick me up after experiments at Loughborough University when we had a budget shoestring, and she was supportive throughout, who has become and blossomed into a phenomenal wife, mother, business owner. She now has a very successful cake bakery business. I forgot to include the pictures there, but thank you to all of my family and all of my parents and my colleagues and collaborators and friends. So final thoughts, and this is my final slide. For me, the challenge is about the manufacture. It's not a scientific challenge, it's a manufacturing challenge. So we need to start thinking about how we can manufacture these therapies at scale. No one can do this alone. And I think that's evidenced by those in the room today, but our teams, our industrial collaborators, our clinicians, our scientists, our partners, we all have a role to play. And only through that collaboration can we actually achieve what we want to achieve. And then finally, the future for me is being built now. The decisions we make today will shape the therapies of the future. The technologies, the processes, the learnings, and the knowledge, and the people will make this a reality. And for me, this is just the beginning. Let's start getting to work. Thank you all very much.
Okay, so I'd just like to add a couple of more thanks. So thanks to all our professional services team, both at faculty and department level, that have organised this event today. So thank you very much. Thank you all. I'll stop there for a moment. Thank you. I would like to thank everyone for attending today, both online and in person. It's great to see a full audience. So I think this is one of our best attended inaugural lectures. So thank you all for attending. And then finally, just thank you for, as I said, setting the bar so high. What a wonderful talk. I think it's an amazing career to date, and it's going to get even better. So thank you very much. Before you applaud, we won't take any questions, but I hope you'll be able to join us outside, where hopefully we can network, mingle, and then ask any difficult questions outside. So finally, thank you, Professor Kasim Rafiq, for such a wonderful inaugural lecture.
Thank you very much.